And the camera is locked on now, so.
Okay. Hi everyone, welcome to the seminar. And yes, as March had said, it was the very first. Can I just close this one? Because
Oh yes.
Yeah. So it is the very first March seminar talk. So during the launch, I already received a lot of pressure from my fellows. Like, it depends on how the first one goes, and then we will see the tendency. So. Yeah, well, I mean, it's not very helpful, right, to make me feel more relaxed before my presentation. And even more, not you, but your fellows, okay? And even more was like, as you can see from the audience, we do not only have the colleagues from mathematical departments, we also have colleagues from biology department and also medical school. So I have a mixed audience, which I would really love to keep everyone awake. But I also keep joking to people, was like, let's see after my talk, whether it's biologists or mathematicians or engineers fall asleep first. Yeah, and so I decided to, because of less complication of the audience, I decided to give an overview of my research, which is definitely not an easy task either, so make my life even more difficult. I suspect I'm not going to finish my talk within the time, so let's see what we're gonna do. So for people who have already talked to me, discussed the research with me, you know that I'm an Asian-based model person, and I specifically work on cell tissue interactions and then the applications, including wound healing, cancer cell metastasis, and also everything basically just related to cell tissue interaction. And another main part of my research is lying on the model upscaling. Which basically how to upscale the model from Asian-based setting and then to the continuum, or we so-called PDE setting. Okay, so for people working on mathematical biology, especially people who are working disease-related, at least I hope we all have the same goal, that we want to apply our model one day in the clinical practice and then help our patients. So in my view, that's the mathematical biology, if you just based on the scale, you can basically have the three different scales, the molecule, cellular, and also the tissue scale. So once you go larger scales, and then you're talking about more cells in the observed domain. And then of course, because you would like to keep your model still computationally efficient, you have to ignore quite some information. For instance, from the molecule scale to the cellular scale, basically we neglected what is happening inside the cell, like the DNA, RNA, and also the protein production. So my work mainly lies in the cellular scale, and then my upscaling work is basically from cellular scale, how to upscale to the tissue scale. There are many challenges in this interdisciplinary field, and these are just the one I have seen so far. And for the first four, I have been working till now, and then since the first day of my PhD life. And I'm also currently slowly entering the field about do the parameter estimation and also have more work about the model validation. There was a very clear time delay here. So in this talk today, I'm going to discuss the application part, which is more modeling. So I guess that part of the room will stay awake. And then secondly, I will discuss more mathematical perspective. That's also the part I have been working on, also till today. And then I also have other side projects, but then probably just a quick glance on what I have done there. So my entire research started with burn injuries. And during the wound healing process, the wound contraction actually is a normal process. So it happens in every wound healing process. But then if this contraction becomes more extreme or excessive, then it causes a very common complication after the burn injuries called contractures. And contractures often happen with the dysfunctioning of the joints. For instance, like the wrist of this patient, because of the skin sticks together, so the patient may not be able to use his wrist or her wrist anymore in the rest of the life. And then So that's why to understand better with the congestions, we actually go back to the congestions. This happens during the proliferation stage of the wound healing, which is known as the third stage, and mainly in the dermis part. And then it's mainly because of the fibroblasts and also myofibroblasts, they exert too much forces and then they pull the skin together, then and then the skin is not able to stretch anymore. So in the dermis part, there are a lot of things happening. So that's why in the modeling perspective, we have a lot of biological components like the cells, cytokines, and the fiber or the collagen bundles. So a different part, how to say, like the distinguishing part for the burn injuries comparing to, for example, the paper cut, or when you're cutting your vegetables, you cut your finger with a knife, is that burn injuries does not have the first stage, where most of the time you will firstly bleed, and then those bloods will make out the clot, and then you do not lose any more blood here. So that's why our model for the burn injuries, we started from the second stage called inflammation. And since we are looking at the congestions, so the proliferations are also considered in our model. So note that at least 4 stages, they are not completely distinguishable. So they're partially overlapping with each other. So actually our model also covers a very small part of the remodeling. And as mathematicians, we do a lot of simplification. So yeah, biology people, please don't feel offended. That is how I build up my model. Is that if you have a wound, we firstly say like the wound is fulfilled with a cytokine called PDGF. And then those are the attractants for the immune cells, which I know there are many immune cells, but let's just say we have macrophages. So the macrophages are attracted by the PDGF and then coming through the broken vessels, which is at the wound edge. And then the macrophages, they will release the TGF beta. So those are also very important cytokines or signaling molecules. They will attract the fibroblasts to migrate from the healthy skin and then to the wounds. And then also because of the high concentration of the TGF beta, they will be triggered to either go for proliferation or go to the differentiation to myofibroblasts. And the main problem for the contracture, excessive contraction, is mainly because of the myofibroblasts, because they have the characteristics of the muscle cells, which means comparing to the fibroblasts, they will exert even larger and significantly larger forces than fibroblasts. And then even though the myofibroblasts, they disappear at the end of the healing process, there will still be some residual forces remained in the wound region. So in this modeling, we include the 3 different types of the cells, and then 3 different types of signaling molecules. I think the TPA, as far as I remember, is also related to the collagen or the fibrin development. And then we determine the displacement of every single cell just based on different mechanics. For example, like the cell-cell interaction, the chemotaxis, which means the cells are attracted to the high concentration of certain chemicals, and the passive convection, because the cells are living on the tissue on the substrate, which means if the substrate is deforming and then or migrating, and the cells are forced to migrate together as well. So that's why it's a passive convection. And then for this part, we use a 2 different approach. One is called plasticity approach, which is more phenomenological. And then the other one is called morphoelasticity approach, which is widely used in mechanics if you want to describe the deformation or the stretching of certain shapes. And then, of course, we also have the random walk here. So for the signaling molecules, we model it with the advection diffusion equation with different types of the reaction term. It depends on what kind of signaling molecules you are looking at. And then for the force balance part, which I meant with the it's the passive convection here. So the work started with the very simple viscoelasticity model because the skin has memory. So that's why instead of using the linear elasticity, we consider the viscoelasticity. And the nice part of this is that it is still linear. So computational wise, it's also more efficient. And because of the residual forces, we split the forces exerted by the cell as one as the temporary force, which means if the cells disappear, these type of forces, they will be gone as well. And then the other one is called permanent force. So these are the ones to model that even though the cells are disappearing from the domain, there will still be some residual forces there. So that's why once the patients develop the excessive contractions, they will have trouble to recover back to the original skin stretch. And then this part is more physical based, and then it was developed by some other PhDs back to 2018, I think. So here for the right-hand side, as the force density term, we still keep the temporary force. But then now to model the permanent deformation of the wound, we introduce this equation, which also solves the epsilon, which is the strain tensor. And then the new parameter alpha determines like how bad or even worse like contraction the wound has after all the cells removed from the wound. And then for the collagen bundles, we use the approach developed by Professor J.D. Dallon and also extended further by Kuming and his collaborators. So basically, we consider here that the ECM, the extracellular matrix, is a continuous media, which I know in reality is not true. And then what we did here is that if it is in two-dimensional, we build up a 2x2 matrix, and then we solve every element of the matrix by this partial differential equations. And then the orientation, and I think also the strength or the length of the collagen bundle is determined by the eigenvector and the eigenvalue respectively. Yes. So thanks to my master student, Wilbur Horton. So he transferred all the model and then from Python to C++. So with his work, we are able to now modeling about, I think in here, around 3,000 or 4,000 cells. And as you can see, the wound area, it will contract first. And then because of the myofibroblasts entering the wound, so it will contract very badly in the beginning. And then after the myofibroblasts slowly dying out, this will go back, recover a little bit. But then because of those residual forces, the wound will never recover to its original shape or volume. We also did some comparison between these two different approaches to model the contraction of the wound. And for now, this comparison is mainly stay in the numerical simulation level. But we are already quite happy with the results in the sense that, for example, if you consider there's completely no contraction, no residual forces, and then these two curves, well, many curves, different color, are aligned with each other quite well. And if you consider that there are still contraction after removing all the cells, for the long-term wise, you still get quite nice agreements, but then here you start to see some discrepancy here. We're not yet very sure whether we can do this type of comparison further from the theoretical point of view. But then this is already a very nice and also promising results that these two approaches definitely have some hidden connections. We also did some sensitivity tests because we have so many parameters, and then we would like to know what are the key factors to influence the contraction. So to be honest, most of them are fitting our expectation. For example, if you know something happening in the early stage of the wound healing, you can kind of predict or link it to what is the final contraction going to look like. or the softer skins, they will suffer more of the contractions comparing to a more stiff skin. But the interesting part coming out is that when we are doing the sensitivity test regarding the immune cells, so for the immune cells, we are mainly talking about the macrophages, which are appearing only at the wound edge, and then we assume there's no proliferation. And regarding like at each time step, how many immune cells popping up in the wound, we use a Poisson points process with parameter not the zero. So initially, I was expecting that it should also have a monotonic tendency. For instance, like the stronger immune cells, the more contraction or the less contraction you're going to have. But this is not what we get. And besides that, I also have another question for myself, but then also for the biologists sitting in the room. How do we define the strength of the immune systems? I know that all three, they make sense, like how many immune cells are in the wound healing, and or how active they are regarding like how many this type of TGF-beta have been secreted regarding time or regardless of the time. So here we mainly play a little bit with the number of immune cells by just twisting this lambda zero parameter. And then what we get is that indeed we do not get the monotonic tendency here. And then we tested both of them. So for this one is that every macrophages, they will produce the same amount of TGF-beta. So you can consider all the immune cells, they are identical. And then for this one, we just say, doesn't matter how many cells you have, but then every time that you, the total newly secreted TGF-beta is the same. And then it's actually kind of matching the clinical observation that I was told by the surgeon from the Netherlands, that what they observed that in clinics, actually to a certain extent, a weaker immune system is better to prevent the contraction, which is exactly what we have seen in this part. And from the simulation, which I'm not sure whether this hypothesis is real or not, but then if the immune system is too weak, which means we don't have enough immune cells to release the TGF-beta, then the TGF-beta is not enough to attract the fibroblasts. However, if you have too many immune cells coming, because they are all popping up at the edge of the wound, so they build up a wall. And then when the fibroblasts want to coming in, they just couldn't enter because they need to squeeze themselves so much, and then it will cause them actually to apoptosis instead of manage to enter the wound and then fix the wound. So yeah, as I said, I'm not sure whether this type of hypothesis is true or not, but yeah, we'll definitely go further with how to model the immune systems better in the wound healing work. So after I moved to Lancaster, it was very nice to meet Richard. So he proposed me a very nice video and regarding the wound closure, or in a more biological way, it's the epithelialization. And so before we are talking about what is happening in the dermis part, which is the second layer of the skin. And then here we are talking about the top layer of our skin called epidermis, which contains several layers of the cells. And I remember back to the time when I was looking at the article, people are still discussing about whether the layers are actually moving down or the cells from the bottom layer moving up and then close the wound. So as some of you probably know, Richard is a person very passionate about the cell cycle. So that's why what we are trying to understand is that what we can do to do something with the cell cycle, and then to understand it better, especially about the wound closure part. This work is collaborated with a PhD student from Uppsala University in Sweden, and he is using a framework which also sitting in the category of the agent-based model called DOCM. So basically, it's kind of an extension of the cellular automata model, but different from the traditional one. Because in the cellular automata model, typically you just only allow one cell occupying one pixel. But then here, every pixel can contain 0, 1, or 2 cells, depending on different cellular events. And so Eric will come to visit us in the week of 10 November. And I think we agreed it's Tuesday, right? Yeah. So he will also give a mass seminar talk on Tuesday. So I will not talk too much about this part of work because he's really the main author for now, like to work on this project. Yeah, thanks to Richard and Leah. So I understand a little bit better about how the things are set up in the wet lab. So basically how Richard and Leah create a wound is that they put something called a PDMS, which I don't know what it is, but this is something the cell really hates. So they don't want to enter this region, which means if you cover some region with the PDMS, when you remove it, you will have no cells inside. So it was kind of like a wound here. And the very nice thing of this experiment is that they just let the cells proliferate and also migration, but then there's no further external stimulus for the baseline model. So Eric did some work by converting what the data that we got from Richard and Leah, and then matching the framework with the pixels. And then here was like either the cell cycle or the number of the cells here. So we have already got very nice promising results, did some qualitative or even quantitative comparison. So we checked the cell counts, and then also because there you have a layer, all the cells moving to one direction. So we are also checking what is the front position is, and then it was already quite nicely matching with the data that Richard and Leah provided us. Yep. Now let's entering the part about the cancer cell migration. So in this work, which is the final project of my PhD, we mainly study how the cell geometry is changing and when they are interacting with the extracellular matrix. And of course, cell does not really have a fixed shape and also the size, and then no need to say the very aggressive type, I mean the cancer cells. And our collaborator on this project, Daphne Weiss, who is a bioengineer, in her lab, they found that actually cancer cell can exert relatively large forces, about 60 nanonewton, which, as far as I remember back to that time, it was not very often that this type of forces are included in the modeling. So in this work, what we are looking at is how the cells collaborate with each other when they need to go through the very narrow channel or the narrow pore, such that the following up the followers can benefit from that. So the idea came from relatively seen popular experiments in biology fields when people are studying how the cancer cell migrates in a confined environment. So basically, they built up this type of device containing all these micro channels made by the glass, because you want the cell to feel the a little bit compressed first to trigger the migration mode. So that's why typically the size of the channels is a little bit smaller than the typical cancer cell radius. And then in Max Lab, they also have this constrictions part, which is even more narrow than the typical nucleus site, which means not all the cancer cell can survive while migrating through here. And then from Peel's lab, we saw apparently they had different types of microchannels, but unfortunately, back to the time, we didn't see any relevant data or results published from Peel's lab. And oh, it was really a strong delay. And then from the mathematical point of view, it's also a very interesting topic. So I have seen even more than that, but then different types of the models just to trying to reproduce such a experiment. In my work, I developed a phenomenological model. What I did is we assume that every cell has an equilibrium shape, which you can understand that when there's no stimulus and then cell is at its most comfortable status. And what we did here is we split the cell membrane into a finite amount of line segments by these nodal points. And then we connect the nodal points not only with the cell center, but then also with the neighboring point by the spring. So that's how the cell will maintain its cytoskeleton. And then how we check the evolution of the cell shape is to check the displacement of every nodal point. And then those are the result of the combination of various mechanics like the chemotaxis, random walk, and then these springs working and the passive convections. The nice thing of this phenomenological model is it has large flexibility regarding the cell shapes. So those are the shapes that I have seen during the simulation, which are quite irregular. And I hope they are realistic as well. So different from the wet lab experiment, because microtubes, they are built by glass, which means doesn't matter how much the cells are pushing, the channels are not deforming. But it's not what is happening inside our human body, because for the ECM and also the blood vessels, they're all soft. If the cell release the forces, and then they should deform, I'll say correspondingly. So we consider two different cases. One is just the single cell migration, which means every cell is at every moment only maximum one cell is allowed in this channel, and then the next cell is only allowed to enter once the previous one completely left. And then the case two is the simplest version of the collective migration. So basically, we just let two cells migrate together. So here comes the numerical simulation if you have only one cell. So the red dot here is as a point source of the nutrition. That's what is the main reason to let the cell migrating from the left-hand side to the right-hand side. So as you can see that the cell is exploring and also going forward, but then in the meantime, because of the forces, the channels are deformed as well. And then for the second one, we just let them migrate together. And then what you can see is that in general, comparing to the first video, they two are already migrating faster. So this is also what we found, and also did the statistical test to check whether that was significant or not. So because the second case, well, the cell doublets. So basically, at some moment, the two cells are migrating together as a whole. So that's why they have more or less the same speed. But as you can see, if we look at the cell one, in case one, if it is just migrating along, it is slower comparing to migrating together. The reason why is that for cell two, it's already wide enough that it can migrate more comfortably, but then cell one is blocking its way. So that's why cell two is also releasing the pushing force such that the cell one is also benefit from that and then migrating faster. We also checked the invasiveness comparing with the single cell migration and then the cell doublet by checking that after each simulation, what is the damage to the channel. And then as you can see for the collective cell migration, the channel width is significantly larger than if there's only one cell migrating. Okay, I still have 15 minutes, and then this part actually I have many to say. Yeah, let's see. So mathematicians can stay awake. Yeah, so the motivation is mainly because there are many cells for wound healing. There are also many models for wound healing, but then there are also many models for the cancer cell. But if you ask me like how many of them have already been applied or even close to be applied to clinical, it was very, very small portion. So my idea would be is that instead of keep developing new models, it's more important to find the connections between the existing models, and then also with this model upscaling, it's another way to improve the computational efficiency in the sense that you're looking at more cells in the tissue scale. So this work has been done with the linear elasticity equation and also the diffusion equation. So because the diffusion equation is the work is still going on, so I will briefly talk about this one. And for the linear elasticity or the momentum balance equation, actually, we already managed to upscale from the spatial exclusion model, from the point source, and then to the cell density model. But it's unfortunately not the straight way. You need to take some detour here. So for the diffusion equation, we're considering the biological motivation regarding if the cell is releasing the compounds through the cell membrane. And then for the spatial exclusion model, basically we split the intracellular and extracellular environments. And to make my life easier, we just remove the intracellular environment out of the computational domain. So only this blue part is the valid computational domain or observed domain. And if you zoom out enough, and then every single cell becomes just one point. And then that's how we end up with this point source model. So basically, if it is a circular cell, you just consider all the compounds coming from this point source from the center and then started diffusing. Yes, and then the method I use is Dirac delta distribution, which I started fighting with it from my day one of my research till now. Yeah, so for the spatial exclusion model, because basically in this computational domain, you don't really have any source term. So the secretion of the compounds is modeled by the inhomogeneous Newman's boundary condition. And then here the phi xt is the flux density at every time step. And then with the point source model, we don't have to do anything regarding the computational domain, but then you just, and then also nicely, it is still linear equation. So you can simply just sum it up and then later on applying the superposition theory. And for the outer boundary condition, we just use the traditional homogeneous Newman's boundary condition, which means nothing coming in and also nothing coming out, going out, sorry. And then firstly, we take a look at whether it is possible to have the same solution from the two models. And if so, what is the condition? And then we end up with this flux condition at the boundary, which the intuitive explanation is if at every time step, at every point over the cell membrane, they are releasing exactly the same amount of the compounds to the extracellular environments, then yes, the solutions are exactly the same everywhere. But then this condition will can never be reached like analytically or numerically. So we started with some simulations, and then we started simple with that. We only consider homogeneous flux density, which means the phi here from the spatial exclusion model here is just S1. And we run the simulations for relatively large diffusion rate and also relatively small diffusion rates. The small and large is defined by, because if you, because for the spatial exclusion model, the diffusion already started from the cell membrane. But if you look at the point source model, the compounds needs to travel from the cell center to the membrane and then from the membrane to the extracellular environment. So that's the reason why that's depending on the relations we pick up between the diffusion rate and the time step, you get some discrepancy here because of this systematic time delay. So when I look at this part, I was like, okay, they look quite parallel to each other, which means if I shift my red curve a little bit to the left, then there's another chance that those two curves are going to overlap nicely as here. And then to translate into mathematical language, it means we can give an extra initial condition for the point source model to compensate this discrepancy part. And we use the fundamental solution of the diffusion equation, which is in the form of the Gaussian kernel. And here we will have two different parameters. One is that how large this concentration thing should be, and then the other one is that how early you want the point source model to start diffusing. And we did some different investigation of how to pick up the P0 and T0, but not going to be discussed here. Okay, so what I'm trying to convince you is that if you indeed plug in the Gaussian-shaped initial condition for the intracellular part, it will catch up better with the spatial exclusion comparing to if inside everything is just 0 initially. Yeah, we also checked the computational efficiency. And then if you look at these two different methods, actually, when you're solving the boundary value problem, they cost more or less the same. But then the main problem is while you are generating the mesh. And then if you want to do the spatial exclusion model, which means once the cell started moving, every time step you need to regenerate the mesh part. Then we started entering the field, the world regarding inhomogeneous flux density, because we are considering a circular cell. So we definitely need a spatial-wise 2-pi periodic function. And yes, the simplest one is sine theta, sine function. And then the other reason is that because of the Fourier analysis, so there's like 1 sine theta, sine 2 theta, etc., they build up a proper subspace. And because of this inhomogeneity, we came up with this multi-Dirac method. So basically, because at the maximum point and also the minimum point of the sine function, those are the largest inhomogeneity. So that's why the extra Dirac source is sliding between this line. Of course, you can also go to this line, and I can promise you that you will get the same, well, not exactly the same expression, but in the end, the flux function, you will get the same. So we go back to our boundary condition regarding the flux to see how we can make the two models, consistent with each other. So for the point source model, we started with the fundamental solution to compute the flux. And just to make the life a little bit easier, so if you can take this part outside of the integral, the entire thing can be simplified into this version, which is quite nicely. And also by sending t to infinity, you get a steady state of the flux. So because of that boundary flux equation, which is written here, you can only approximate it, but then not make them equal. Then the question becomes how you approximate it. So I started having this wild idea, was like, when I was doing my calculus, the extremum points are very important. So how about just enforcing this point and this point, this part hold? And then I end up with two equations with two unknowns, which are the intensities of the Dirac delta sources here and also here. And after just solving it by hand, even, you get this two expression. And then also by sending t to infinity, you got this stable value. So we also did some parameter values and then taken them from one experiment paper. So that's how we decided the parameter values in our simulation here. And we checked the flux density to see whether our idea about the extremum points working or not. What you can see is that if you look at the flux at the infinity time, and then once you reduce the r, which is the distance between the two Dirac delta points, you can see that this curve is getting closer and closer to the sine curve that we want. And then the same thing, the same thing happened that if you chose a relatively small enough R, by sending the time t to larger, this curve is also moving closer to the sine curve. And then we are already able to prove that by sending t to infinity and also r to 0, then you can get the sine curve back. So both of the Dirac sources, they are, so for sure that the extra Dirac source, it needs to be a source. So it releases the compounds. But then for the center one, it can be a source or it can be a sink, which means it's taking up the compounds instead of like releasing. And then this depends on what is the distance between the two Dirac delta points. Yeah, and here are the norm comparison results. And as you can see, again, if you chose small enough, and then the difference between these two models are actually smaller if you don't do anything, just put one single Dirac in the cell center. And there's a problem with this part is because, as you can see from the shape, when t is very, very small, the intensity can bump to very large value, and then it will cause the failure of the numerical scheme. So we use the singularity removal techniques by splitting the boundary value problem, the solution we're looking for, to u hat and v, where u hat is the fundamental solution of the diffusion equation, and v is the correction term, which can be solved by here. So as you can see, then here is a very nicely defined diffusion equation, and then all the singularity hidden in the u hat has been transformed to the computational boundary. But this one can only delay the occurrence of the blow-up solution, but then not prevent it. But this is not the main aim of our work for now, so that's why we did quite some simple and also quick Monte Carlo simulations, only checking at the diffusion rate and also the amplitude of the sine function. And then it seems there's a very clear line here with the diffusion less than 0.01, and then the simulation will just fail. The solution will blow up. Yes, we are entering the final part. So I have another work which is studying the traveling wave in the wildfire, which has already been published as a conference proceeding. And we, from the analysis, we did the simulation, and then you can see how the wind and the traveling wave is how the connection is. And then I have another part of work which about the focus, the ultrasound and microbubbles for the drug delivery. So we were mainly looking at this Rayleigh-Placid equation because this work is ongoing and my collaborator never wants anything split out, so I cannot show any results here. Yeah, the conclusions. I only have one sentence to say. If you want to know more details and then want to know more about my research, just come to talk to me. And last but not least, I would like to advertise a little bit the study group Mathematics with Industry in the Netherlands at the final week of January next year. So the reason why abbreviation is W is because it's wiskunde in Dutch. This is a very nice event, and then this is our new Faculty of Science of the building. It's very nice. I joined this event 3 years ago and then definitely recommend it. And for the academic participants, there's no registration fee. And well, actually, I want to say definitely the accommodation and meals will be covered, but I checked their website. Every year they say it was like limited number. But I have never heard anyone like failed to reimburse it from the committee itself. So if you are interested or you would like to send some of your students or even master students to this event and then would like to know more, please do feel free to talk to me. Yeah, of course, I would not be able to go this far with my research without my collaborators. And so Sander Hiele is one of my postdoc advisors from Leiden University. He will come to visit me in the week of 17 November, and will also give a talk about the work he had with biologists regarding the plant cells. And as I mentioned earlier, Eric will also come in the week of 10th November. So yeah, stay updated with our Mars seminar. And if you want to see more of my work, etc., you can go to my personal website, and then it has the full publication list. So yeah.
Thank you. That was a wonderful talk. So we open the floor to questions. Does anyone have any question? Please raise your hand.
Yeah.
More.
I have a question. What's the intuition behind those permanent forces that stay up, that the wood can stay up and cells?
Yeah, it was because like even though the cells are dying, the residual forces are still there. So that's why it created the tension and also the stress, and then keep pulling the forces. Because the complication, the contracture, it will only get worse and worse. So which means actually the forces are still just keep applying on the. I don't know. I think it's mainly because the skin has the memory to some extent. So that was how I see this as a mathematician.
Any other questions? Thanks.
You had results where you explained the condition on the flux, so that the spatial exclusion model has the point source model, and then you were trying to potentially correctly have the two models be approximately the same, and this was numerical. Is there a theoretical result that says was actually the results can be exactly the same, or it can never be the same?
Sorry, No, so for this one, we have a rigorous proof for this proposition. So how you can see is that the two models are converging to each other to some extent, but then this condition it will never be able to reach. It's just purely because physically, for the point source model. As you can see that the compounds started the travel from here to the membrane, while in this model, everything already started from the membrane itself. So you always need, the compounds always need a physical time to cover from this part to this part.
Okay, so you mean to say you can reach it in practice, but theoretically?
Sorry again?
So you're saying in practice, such inequality wouldn't be opposed, but in theory you could have it? Is that right?
Yeah, so you can see from the condition here, it's also no. So I would say those two models are only be able to give exactly the same solution if and only if this condition can be satisfied. And so that's why when we're doing the analysis, etc., we always try to start here and then to make this two as close as possible. Yeah.
Yeah, just a more general question. Are the models saying anything about how you might go and set up an experiment? Kind of maybe measure what goes on during the mood feeling and then kind of solve the inverse problem, try and work out the parameters that you've chosen to drive the model progress?
Yeah, so it was a very hard question, to be fair, because what I am doing with agent-based model is more corresponding to wet lab experiments. But then if you are talking about disease, of course, you are talking about clinical practice, and those two data are completely different. There is connection. For instance, if you as the age increase, and then the stiffness of the skin is in general change. So that's why I think there's definitely possible to find the parameters if we can have some wet lab experiments. But then with the clinical data, you can only build up the very vague connection. For example, if you're increasing the age, or if you are changing the gender or the race, which certain parameters you also need to tune respectively. And then for the wet lab experiment validation, that was already the work going on with Richard and also Eric and also Leah.
Yeah, I was talking about the scope for personalized medicine. So somebody had a bed sore also.
Yeah, so that is also the reason why I find the model upscaling quite important, because then from agent-based model to the PDE, if you can successfully do the upscaling, you can also find the connections. For example, from cell count to cell density, how you can pick up the unit volume or unit area. Yes.
I was just wondering, the model you use, do you see a difference if you change the shape of the wound that you use it? Probably like the rectangular wound. If you have a different shaped wound, do you see a difference in your mechanics? And is there like difference in any aggregation of cells in the
I know in the reality it will change the things, but currently my model is not able to capture that. Because when you talk about the shapes, you are also talking about, for example, the curvature, etc. Those are also that will influence how the cell will, how the wound will heal. But then currently in my model, I didn't consider the curvature of the wound. But then that was also one of the many things I want to do to extend the model. So, thanks.
I was wondering how you parameterize the channel for it to react to the cells. What happens? How does it degrade? Or yeah.
Yeah. So what we did here is that we just consider the channel is there on the substrate. So what you see how the channel is moving is that at that point of the substrate, there was the displacement as a result of the, so the channel itself does not have any resistance or any mechanical function. And yes, I know that one.
It takes that's a force on the cells, which allow the second cell to change shape differently. Also, yeah. Okay.
Yeah, I have also a general and potentially naive question, which is about how do you, I mean, I think partly related to what Chris asked, how do you validate your models? And partly also how you parameterize it, and are you mainly after qualitative behavior? And if not just qualitative behavior, what is stopping you to reduce quantitative behavior? So is it like a limited amount of data? Or so for example, like I mean, one natural thing is you mentioned like if you have like movies or time-lapse movies of wound healing, with the agent-based model, you could potentially like parameterize your agents fully in a data-driven way. But I don't know.
Yeah, so that was a many bunch of questions.
Yeah.
Let me recall the first one. What's the first one?
How do you validate it?
Okay, so for the validation, for the wound contraction one and also the cancer cell migration one, they're only no, the cancer cell one is not even validated, because back to that time when I do that, we searched all the literature in the lab experiment. No, they only build up the channels with the glass. So basically, we don't have any data which is possible to do some validation. But then the idea itself, so for example, in Mark's lab, they publish some results considering non-deformable microchannels. So that one we did some validation with I think it's even quantitative validation for the model. For the wound contraction one, it's mainly the qualitative validation regards like we read different literature. For example, they say when you increase certain things and then certain things drop by certain percentage, etc. So doing in this way. What's the other questions?
In the interest of time, because it's, yeah, we should talk later. I mean, we can always talk later. So let's thank Alice again. Okay, well, thank you for turning up. It will be a difficult thing to keep it going with these numbers, but we'll see. So the seminar next week from Andrew Buckley, who's our new Mars professor. So yes, see you next Wednesday. Thank you. Very good.